Hello, welcome to Lab 3 of Cell and Molecular Biology. In this lab, we will study prokaryotes, which include bacteria, and cyanobacteria, which are a type of bacteria. We'll first start with bacteria. We will use a technique called gram staining, which allows us to see different kinds of bacteria and their shapes. So different bacteria have different kinds of cell walls. So some bacteria, for example, have really thick cell walls outside their plasma membrane or cell membrane. And these bacteria tend to take up and keep a lot of stain, and therefore they end up looking purple when you do gram staining. And these are called gram-positive bacteria. There are other kinds of bacteria that don't have such a thick cell wall layer, but they have a thin layer of cell wall, and instead of one, they have two cell membranes. And because they do not have a thick cell wall, they cannot keep the purple stain, and so they end up looking different. And these bacteria are called gram-negative bacteria, and they are characterized by a thin cell wall. Now, this cell wall is made up of polysaccharides, which are sugars in chains that are cross-linked with proteins. These polysaccharides are called, together, these polysaccharides with proteins are called peptidoglycans. So the cell wall in bacteria is made up of peptidoglycans. Using gram staining, you can distinguish between different kinds of bacteria. So the thick-walled bacteria are gram-positive, the thin-walled bacteria are gram-negative. This is another nice visualization of the cell wall structure. So if you notice, this is in gram-positive bacteria. They have a really thick peptidoglycan cell wall layer, several layers of peptidoglycan. Whereas in gram-negative bacteria, uh, you have a thin cell wall layer. That's the plasma membrane underneath the cell wall in gram-positive bacteria, and the plasma membrane in gram-negative bacteria. And that's in gram-negative bacteria, in addition to the inner membrane, it has a second membrane which is called the outer membrane. So once you do gram staining, if the bacteria are thick-walled, they keep a stain, a blue stain. That's why they end up looking this way. That's what makes them gram-positive. The other bacteria that don't have a thick cell wall, they don't pick up the blue stain, and they end up looking pink, and these are called gram-negative bacteria. And gram-negative bacteria, in addition, also have an extra layer which is called LPS, or lipopolysaccharide. So the gram staining process goes through several steps. So in the very first step, you take bacteria and you attach them to microscope slides in a process which is called fixation. So you put some bacteria on the slide and then expose the slide to heat, and the heat fixes the bacteria to the slides. So if you have gram-positive bacteria or gram-negative bacteria, they can both be fixed using heat. The next step is you add crystal violet, a blue dye called crystal violet, which then stains all the cells blue. Now the next step is to add iodine. Iodine is referred to as a mordant, which is the purpose of iodine in gram staining is to attach to crystal violet. So it forms a complex with crystal violet and makes the crystal violet insoluble by water. So in other words, it helps the cells retain the crystal violet. After iodine treatment, you do a step called decolorization, in which you use ethanol or alcohol. So when in gram-positive cells, when you use alcohol, the it, it does not decolorize them because it does not remove the blue stain because of the thick cell wall. The thick cell wall prevents the stain from getting washed away by the alcohol. On the other hand, in gram-negative bacteria, you have a thin cell wall layer and you have an outer membrane. The outer membrane gets dissolved by the alcohol, and the thin cell wall does not block or prevent the iodine crystal violet from going away. So at the end of decolorization, the gram-negative bacteria become colorless. Now, after this step, when you add a counter stain or a second stain, which is called safranin, safranin is a pink stain. So when you add safranin to the blue crystal violet, then you end up getting a purple. Blue plus pink equals purple. But in gram-negative bacteria, the cells became decolored. They lost their color during the decolorization, and when you stain them with safranin, they end up looking pink. That's the reason why gram-positive bacteria show up as purple cells after gram staining, whereas gram-negative bacteria uh, appear pink after gram staining. When you do staining for bacteria, you can also um, tell their shapes. So some bacteria, there are three major shapes overall. Some bacteria they have circular or spherical cells. Some other bacteria have rod-shaped cells. So they may be small or long rods, and other bacteria have spiral cells. So some examples of these bacteria, um, there's a streptococcus or streptococci that this is the one that can cause strep throat. And there is staphylococcus or staph, simply called staph, and this is a common skin bacterium. And so in this case, in streptococcus, the cocci or the circular cells are arranged in chains, whereas in staphylococcus, they are in clusters like a bunch of grapes. In the rod-shaped bacteria, there are different kinds of rod-shaped bacteria. One example is Pseudomonas. Pseudomonas is one of those bacteria that can live in the lungs of uh, patients with cystic fibrosis and causes all kinds of problems. So those are rod-shaped bacteria. Then you have the spiral bacteria. One example is uh, the treponema, that these bacterial cells look really different. And you can also see sometimes the cells are arranged in a certain pattern. For example, they are in chains, in some cases like chains of circles or chains of rods. In the second part of the lab, you will look at cyanobacteria. So as I mentioned, cyanobacteria are a kind of bacteria. They're a group of bacteria. And while bacteria are colorless and you need a stain to visualize them, the cyanobacteria are already colored because they tend to have pigments like photosynthetic pigments like chlorophyll, for example. Cyanobacteria, you can observe them in nature, and sometimes they cause all kinds of problems. This is Lake Erie, and this is a satellite image, and you can see cyanobacterial blooms. So where they grow like an outbreak. So this is a large bloom in the West Lake Erie a couple of summers ago, and this is basically all of that bloom, and this was triggered because of agricultural runoff. So if fertilizers from nearby fields get in the groundwater and they end up in the lake, then they promote the growth of cyanobacteria. And so some cyanobacteria, like microcystis, they can be toxic. They when they overgrow in the water, they can deplete the oxygen, and they can also release toxins called cyanotoxins that can kill animals like fish that live in the water. And if the water from the lake, when it gets through the purification system and it's not adequately purified, then it can also cause problems for drinking water. There are other kinds of cyanobacteria which are not necessarily bad, and actually they are really important ecologically. There is one which is called Anabina. You can tell Anabina from the shape of their cells. They have a bead-like appearance, so it's like a string of beads. And Anabina, one of the most special characteristics of Anabina is it has the ability to fix nitrogen or nitrogen fixation. So nitrogen is the most abundant element in the air, but it's really hard to use by living organisms. But Anabina, in those special cells, has some special enzymes that can actually uh, use the nitrogen and converts it into a usable form, which most other species on Earth cannot do. Then there are other cells which are larger and thick-walled, and these are called akinetes. These are stress-tolerant cells, so cyanobacteria make these during times of stress, and they are protective and they can help overcome the stress. So you can see a heterocyst, which is not much bigger than the other cells, and then you can also see an ekinete, which tends to be larger than the other cells. The last cyanobacterium you will look at in the lab will be Oscillatoria. So Oscillatoria is so called because the cells exhibit a, oscillatory motion, so they oscillate towards light. And as you can see, these are green, so they are photosynthetic, so they seek out light for photosynthesis, and that's why the motion. And so basically, you'll be looking at three cyanobacterial samples. One is Microcystis, the second is Anabina, and third is Oscillatoria. All right, see you in lab.